last videos we have studied about the formation of the male gamete, female gamete, and how and transfer of pollen grain, the transfer of the male gamete to the female gamete, transfer of pollen grain from anther to stigma, pollination we have studied. So in this video we are going to study about the pre-fertilization events, fertilization events, and post-fertilization events. So the pre-fertilization events we have studied, the formation we have studied, and also the transfer we have studied. Here the interaction of the pollen grain with that of the ovary or the pistil we are going to study, and after that one what are the post-fertilization events also we are going to study. That is the formation of the seed, zygote, then embryo, all those things we are going to study in this video. So first topic which we are going to deal here is pollen-pistil interaction. Okay, so in pollen-pistil interaction, now the male gamete has formed. Male gamete has formed in the sense from the anther, the dehiscence of the anther, the release of the pollen grains takes place at 2-cell stage, isn't it? At the 2-cell stage, the release of the pollen grain takes place at the 2-cell stage. Okay, this is the vegetative cell, and this is the generative cell, isn't it? So at this 2-cell stage, the release of the pollen grains is going to occur. Sometimes what happens, this generative cell is going to divide into two and form into 2 male gametes. At this 3-cell stage also in some plants takes place. The majority of the plants at 2-cell stage the pollen grains are released. In some plants 3-cell stage it is going to release. 3 cells are formed from the generative cell, because the generative cell is going to develop into 2 male gametes. Okay, so now the pollen grains are released. They have fallen onto the stigma. So if they have fallen to the stigma, the pistil should be in such a position to identify the correct one, right one, and accept the right one, and reject the wrong type. Okay, so the compatible pollen grains must be accepted by the stigma, by the pistil, and the incompatible pollen grains must be rejected by the pistil. The stigma has that capacity. So the pollination does not guarantee the fertilization. Okay, does not guarantee the fertilization, because sometimes the wrong type of pollen grains also fall onto the stigma. So the pistil should have the capacity. The pistil it has the ability to recognize the pollen grains of the right type, compatible type of pollen grains, and then accept those compatible pollen grains and reject the wrong type, incompatible pollen grains. So if the compatible pollen grains are accepted by the stigma, that particular pollen grains are going to germinate. They are going to germinate and develop into and they're going to develop into the pollen tube. And that pollen tube is going to have the male gametes. So what happens, so here the pollen grain, there will be a dialogue between the pollen grain and the pistil. There will be a dialogue, there will be exchange of the chemical components, such that the pistil the stigma identifies the compatible pollen grain, and if it is a correct one, compatible pollen grain, then it is going to germinate and produce the pollen tube, and this pollen tube is going to bear the 2 male gametes. Okay, so there is a dialogue between the pollen grain and the stigma. There is a dialogue between the pollen grain and the pistil, and there will be the interaction between the pollen grain and the pistil with the help of the chemical components. Okay, so now what happens? So after the compatible pollination, the pollen grain has fallen onto the stigma. They are compatible. The pollen grain germinates. The pollen grain germinates means the pollen grain is covered by 2 layers, and at some places the exine is not deposited. The outer layer is not deposited. It forms into a germ pore. So through that germ pore, the pollen tube comes out. Okay, the pollen tube is going to come out. At some areas the exine is not deposited. This is called as a germ pore. So here, when the pollen grain germinates, when the 2 cells, when the pollen grain germinates, it develops into a pollen tube, and then this will be disappeared, and this generative cell is going to divide by mitotically. It will develop into 2 cells. So this pollen tube is going to have 2 male gametes which are formed from generative cell. This vegetative cell supplies nutrition to the generative cell, and after some time, when the pollen tube is formed, this generative cell is going to divide into two cells with the help of mitosis, and two male gametes will be entering into the pollen tube. This is at the 2 cell stage. In the 3 cell stage, what happens? Already the cell will be 2 cell stage. What happens? The registered cell disappears, and the generative cell has to get into together. So these 2 cells are going to develop into male gametes. That pollen grain is going to carry the 2 male gametes in 3 cell stage. Okay. So the pollen grains are released at 2 cell stage and 3 cell stage. 2 cell stage, after the formation of pollen tube, this generative cell is going to form into 2 male gametes. In the 3 cell stage, already the male gametes are present in the pollen grain, and then it will also develops into the pollen tube. Okay. So this pollen tube is going to travel through the style and reaches the ovary. It is going to travel through the style and reaches the ovary. So if you take the pistil like this, this is the ovule, isn't it? Like this. So here the compatible pollen grains have formed. They have developed into the pollen tubes. Okay, they have developed into the pollen tubes like this, and this pollen tube will be traveling through the style and it will enter into this ovary and it will enter into the embryo sac. So that entry of the pollen tube into the embryo sac is of 3 types. Okay. The first one is porogamy. When the pollen tube enters through micropyle. Micropyle is the pore of the ovule, isn't it? So through micropyle, such type of entry is called as porogamy. Okay. So take for example like this. Okay, this is the nuisance like this. So when the pollen tube enters through the micropyle, so this is the micropyle, no? This is called as porogamy. When the pollen tube enters through the integuments, it is called as mesogamy. Okay, when it is entering through the integuments, it is called as mesogamy. So when the pollen tube is entering through the integuments, like this, when the pollen tube is entering into the embryo sac through the integuments, it is called as mesogamy. So when it is entering through the chalaza, it is called as chalazogamy. Let us draw the diagram for that. Simple. Like this. Okay. When the pollen tube is entering into the embryo sac through chalaza, it is called as chalazogamy. This is about the pollen-pistil interaction. So the germination of the pollen grain, development of the pollen tube carrying the pollen grains, and how those pollen tube carrying the male gametes, and how those male gametes are entering into the embryo sac through porogamy, mesogamy, chalazogamy. So all these things comes under the pollen-pistil interactions. Now what is there? The pollen tube is carrying the 2 male gametes. And this pollen tube, it comes near the filiform apparatus, and this filiform apparatus, which is present near the cervix, it guides the pollen tube it guides the pollen tube to enter into the synergids synergids. So the filiform apparatus which are present at the micropylar end, here they are going to guide the pollen tube to enter into the embryo sac. Then what happens? That is about the Pollen-Pistil interaction. Now coming on to the double fertilization. So now the pollen tube has entered into the embryo sac carrying the male gametes. Then the pollen tube is going to break. Okay, now double fertilization. Now we are going to deal with double fertilization. Okay, then the pollen tube, here what happens, after entering one of the synergids, the pollen tube is going to burst and release the two male gametes into the cytoplasm of the synergid. One of the male gametes will be moving towards, let us draw the diagram for that. Okay, this is the embryo sac. Okay, this is the filiform apparatus. These are the synergids, egg cell, polar nuclei, central nucleus. These are the antipodals, isn't it? This is the micropylar end. So at the micropylar end, two synergids are present and the egg cell is present, isn't it? This is the filiform apparatus. These are the filiform apparatus. These are the synergids. This is the egg cell. These are nothing but the egg apparatus. This we have studied already, okay? Egg apparatus. This is the polar nuclei or central nuclei. These are the antipodals. Okay, so what happens? The pollen tube is going to enter. This filiform apparatus is going to guide. It will enter into one of the synergids, and it will be releasing two male gametes. Okay, two male gametes, and one of the gamete will be fusing with the egg cell. One male gamete will be fusing with the egg cell. It results in the formation of zygote. Then another male gamete will be entering into the polar nuclei, central nuclei. Another male gamete will be fusing with the polar nuclei, resulting in the formation of primary endosperm nucleus. So the male gamete, when it is fusing with the egg cell, such type of thing is called as syngamy, or the fertilization, syngamy. Here another male gamete is again fusing with the polar nuclei. These are haploid nuclei, which are 2 in number. So what is happening? 1 plus 2, triploid is formed. So this particular thing is called as triple fusion. So this particular process is called as triple fusion. As 2 male gametes are there, 2 fusions are occurring, such type of fertilization is called as double fertilization. So in angiosperms, double fertilization is common. Okay, double fertilization is common. Then this primary endosperm nucleus is going to develop into endosperm, and the zygote after several divisions is going to develop into embryo. So we are going to deal now with the post-fertilization events. Now with the post-fertilization events. That is zygote formation, embryo formation, then formation of the seed, everything we are going to study. First we will study about the endosperm, formation of endosperm. So this endosperm is formed as a result of triple fusion. It is a triploid tissue. Okay, it is a triploid tissue. This endosperm development is going to form before the seed development. It precedes the precedes the embryo development. Before the embryo development only, the endosperm development is going to occur, because it's a nutritive tissue. It has to provide it has to be formed first, then it will provide nutrition to the embryo anamata. So that's why this endosperm development happens first, then the embryo develops. Then it is a triploid tissue. Yes, it's a triploid tissue. Then it is a nutritive tissue, and this particular primary endosperm nucleus will be undergoing several nuclear divisions, which are called as free nuclear divisions. Free nuclear divisions. After the free nuclear divisions, then the cell wall formation will be taking place and develop into cellular endosperm. But this particular free nuclear division and cellular endosperm is, it can be clearly seen in the coconut. Where the liquid is nothing but the liquid endosperm. Free nuclear division. Then afterwards, what happens? Whatever the white color portion is nothing but the cellular endosperm, where the cell wall formation has taken place. But here the liquid is nothing but it is having the free nuclei present in the liquid amount. So whatever the water and the white color portion is there, that is nothing but the endosperm. It is a nutritive tissue for the embryo. It is a triploid tissue. Endosperm development takes place first, then first endosperm will be formed, then embryo will be formed. Then this particular endosperm, sometimes in the seeds, the endosperm will be consumed by the seeds. So before the seed maturation only, this endosperm is consumed. Such type of seeds are called as non-endospermic seeds, where inside the seeds we cannot see the endosperms. Inside the seeds we cannot see the endosperm, because already it is consumed by the embryo. So non-endospermic seeds. Non-endospermic seeds are many actually. Generally examples like pea, groundnut, bean are the non-endospermic seeds. Endospermic seeds are means where we can see the endosperm inside the seeds. We can observe after the seed maturation also, we can observe the endosperm. The endosperm is persistent. Like for example, in monocotyledons, and you can also observe in castor. Means after the seed germination also, seed maturation also, we can observe the endosperm. In coconut also. Already that particular seed has been germinated, but still we can see the endosperm. Means the endosperm is persistent. This is about the endosperm. Coming on to now embryo. Embryo is formed from the zygote. The zygote is going to undergo several divisions and forming into embryo. So the early stages of this particular embryo are common in both dicots and monocots. Dicots means the seed bearing 2 cotyledons. Monocots means the seed bearing 1 cotyledon. So the early developmental stages of the embryo are same in both dicots and monocots. Okay, embryo. Now we are going to study in detail about embryo. Developmental stages of the embryo, that study is called as embryogeny. It is formed by the fusion of male and female gamete. When they fuse together, develop into zygote. So embryo is formed from zygote. It is going to form into, the zygote is going to transform into embryo. Okay. And this particular embryo is going to be present at the micropylar end of the embryo sac. Okay. So first it is this particular embryo is proembryo. Then it is going to develop into globose embryo. Then heart-shaped embryo. Then mature embryo. Then we can see the mature embryo. These are the early stages. These are common in both dicots and monocots. The dicote is going to develop into proembryo, then globose embryo, heart-shaped embryo, and mature embryo. Then at this stage, it will get get transformed into either dicot or a monocot. Okay, so let us see the diagram for this. So the diagram for this, the pro-embryo, simple. Suppose the zygote is like this. Okay, this will undergo some divisions and develop into the pro-embryo. Like this, 2 cells will be present, pro-embryo. Then globose embryo. Globose embryo means a globular embryo. Okay, globular embryo means like this. Like this. Okay, this is globose embryo. Coming on to heart-shaped embryo. Heart-shaped embryo is somewhat like this. Like this. Okay, it is going to show the divisions. The cells are going to be there. So this heart-shaped embryo. Then mature embryo. The mature embryo, this like this. This is the mature embryo. This will get transformed into either dicot or a monocot. So let us study now. Let us study about the first about the dicotyledonous embryo. Dicotyledonous embryo. Okay, means the seed which is having 2 cotyledons. In that seed, how the embryo is there, we are going to study now. So this particular dicotyledon is going to have two parts. Okay, 2 parts. It consists of 2. That is embryonal axis and cotyledons. Embryonal axis and cotyledons. So if you see the diagram for this, this is the diagram. Okay, this is the diagram. Okay, so these are the 2 cotyledons. This is the plumule. This is the radical, root tip, root cap. Okay, like this it's going to be there. Okay, so these are the 2 cotyledons. This is the embryonal axis. Okay, the upper part of the embryonal axis, the upper part of this is the whole embryonal axis. The upper part of the embryonal axis will be developing into the plumule. It is the early shoot tip. This plumule is going to develop into the whole shoot system, stem. Then the lower part of the embryonal axis is going to develop into the radical. Radical is nothing but the early root system. This radical is going to develop into the root system. Then the tip of the radical is having the root tip, and the root tip is covered by the root cap. This is about the dicotyledonous embryo. And here the upper portion. So if you take this is the embryonal axis now. The upper portion is called as epicotyle. The lower portion is called as the hypocotyle. Okay, so the upper portion of the epicotyle will develop into plumule. The lower portion of the hypocotyle will develop into the radical. So these are the terms which are given to the embryonal axis. The portion of the embryonal axis above the level of the cotyledons is called as the epicotyle. Above the level of the cotyledons. So 2 cotyledons are like this, epicotyle and hypocotyle. This is about the dicotyledonous embryo. If you take the monocotyledonous embryo, it is going to have embryonal axis and cotyledon, but only 1 cotyledon. So here only 1 cotyledon. Okay. So like this, if you take, this is 1 cotyledon, and it is going to have like this. This is the cotyledon. One cotyledon. Okay. Here the difference is that this cotyledon is called as scutellum. Okay, this cotyledon is called scutellum. And the embryonal axis here, these are present side to side. The cotyledon is somewhat lateral to the embryonal axis. Then at the lower end, the embryonal axis has the radicle. It has the radicle. Then the root cap, and the root cap is covered by a special structure called as coleorhiza. This is important. Okay, coleorhiza. Coleorhiza is going to protect the root cap. Coleorhiza. Coleorhiza is the undifferentiated sheath which is going to enclose the root cap. Then the epicotal portion, which is bearing the shoot apex is covered by the another sheath called as coleoptile. Okay, the coleoptile. The coleorhiza bears the encloses the root cap. The coleoptile is going to protect the shoot apex. The protector is going to protect the shoot apex. So here no radical and pure. Here it is the epicotal and hypocotal. It is having the coleoptile and the coleorhiza. And the covering of the embryo, covering of the outer covering of the endosperm. The outer covering of the endosperm is called as the alluron layer. Alluron layer, which is highly proteinaceous. And these monocotyledon seeds are going to have embryo. That outer covering of the embryo is called as alluron layer. The cotyledon of the monocot is called as scutellum, and the root cap is enclosed in the coleorhiza, and the shoot tip is enclosed in the coleoptile. Epicotyle has a shoot apex and a few leaf primordia enclosed to a hollow foliar structure called as the coleoptile. So this is about the monocotyledon's embryo. Coming on to the seed, we are going to study about the seed, dicot seed and monocot seed. It's very simple. Dicot seed is going to have two. Dicot seed. Here the seed is covered by an outer covering called as seed coat, and this seed coat is having two covers: the outer testa and the inner tegment. So these are formed from the integments of the ovule. Then hilum. Hilum is the scar. Hilum is a scar on the seed coat. It is a scar on the seed coat which is going to attach to the fruit. These seeds are enclosed in the fruit, kada. So this hilum is a scar which is going to attach to that of the fruit. It is present on the seed coat. Then above the hilum is nothing but the micropyle. Above the hilum is the micropyle of the ovule. Then these cotyledons are very much fleshy and they store the food materials. And the two embryonal axes are present. The plumen which is going to develop into the stem, or you can say shoot. The radicle is going to develop into the root system. Okay, the stool system. And endosperm. Endosperm is a nutritive tissue for the embryo. Endosperm is a nutritive tissue for the embryo. And sometimes some mature seeds are going to have endospermic seeds. Sometimes some mature seeds do not have the endosperm. They are non-endospermic seeds. We have discussed earlier. Monocotyledon seed. Monocot seed. Only one cotyledon. Which is called as scutellum. It stores the food materials. Then it is all monocots are going to have the endosperm. They are endospermic seeds, and that endosperm is covered by alluron layer. Then here also plumule and radicle are very much there. The plumule and radicle are enclosed in the sheaths. they in dicots it is there, in monocots also it's there. Okay, careful, okay, careful. In both plumal and radical are there. In both monocots and in dicots, plumal and radical are there. But in monocots, the plumal is covered by a sheath called as coleoptile, and the radical is covered by undifferentiated sheath called as coleorhiza. So that coleoptile, coleorhiza are present in monocot anamata. But in both dicots and monocots, plumal and radical are present. Plumal develops into shoot, radical develops into root. But coleorhiza and coleoptile are present in monocot seeds. Coleoptile and coleorhiza in a monocot seed. Okay. Then coming on to the seed. Now coming on to the seed dispersal. So seed dispersal, what happens? See this seed dispersal, how it is. First of all, seeds are produced for water to produce into a new plant, isn't it? So these seeds from the fruit, they must be dispersed. Sometimes what happens, the fruit, like for example in coconut, in coconut it is going to float in water and the seed dispersal takes place. In some plants like in guava, the fruits are eaten by the birds, and in the fecal matter the seeds will be present because this is undigested matter. So that fecal matter is going to deposit on the soil and develop into new plant. Like that the seed dispersal takes place. Then in Martina, what happens? The seeds are going to be attached to the animal fur. Then seeds of guava eaten by birds and not digestible, but the hard seed coat is partly softened. So like that the fruits attract and also the fruits are going to attract the animals by providing food for the dispersal of the seeds. So like this the seed dispersal is going to take place. Then smallest seeds are present in orchids. The smallest seeds are present in orchids. Orchids are the non-endospermic monocot seeds. And they are the microscopic seeds. And the largest seed is present in a Laodicea. Smallest are present in orchids and largest are present in Laodicea maldivica. It is called as double coconut also. It is called as double coconut. Largest seed, okay, maldivica, Laodicea maldivica, double coconut. Largest seed is present. It is somewhat 30 cm long and 90 cm in circumference and width. It is the largest one. And the hard seed coat is there. No, it is going to provide the protection to the young embryo. The seed coat is going to be hard, and the seed coat protects the embryo. And these seeds are nothing but these are formed from the fusion of male and female gamete, which are of the same plant, which are of the different plants. So it's some genetic combination is there. Variations are going to occur that to develop into the different varieties of plants. So variations are going to occur, and the seed is the basis for our agriculture. So seeds are very important. So now the seeds are formed, and when these seeds they fall onto the soil, if the favorable conditions are there, they are going to germinate, they are going to come out, and the radical is going to come out, develop into root system, then plumal, and develop into a new plant. So like this, the seed germination is going to occur. Sometimes, so that is about the normal way of formation of a new plant. That's a normal general of sexual reproduction. How the plant is producing the gametes, how the fusion is occurring, and after fertilization, what are the events are going to occur, and how the seed is formed, and how the seed is going to germinate. Like that we have studied. Now a few techniques are there, like for example Apomixis. Apomixis is a technique where where the seeds are formed without fertilization. The seeds are actually the seeds are the products of fertilization. Okay, the seeds are the product of fertilization. But here the seeds are formed without fertilization. The seeds are formed without fertilization. That is apomixis, especially in Asteraceae and grasses. Asteraceae and grasses. So seeds are formed without fertilization. And another technique is there which is called as Parthenocarpy. Parthenocarpy, where fruits are formed without fertilization. Without fertilization. The fruits are formed without fertilization. Then here no seeds are present. Very, very good example is banana and grapes. The fruits are formed without fertilization and they have no seeds. Banana and grapes are the examples for the parthenocarpic fruits. It can be natural, it can be induced. These play a very, very important in the commercial production of these seedless fruits. These are seedless fruits. So coming on to apomixis, how the seeds are formed without fertilization? That means the seeds may be formed from any part. It can be a diploid egg cell, a diploid egg cell where it will not undergo the rhizomeiosis. Okay, the diploid egg cell is formed. The diploid egg cell is formed without reduction division and develops into the embryo. So the diploid egg is formed without the reduction, without the meiosis. The egg cell is formed, hence it is diploid. So that diploid egg cell will be forming into a mature embryo. So in that sense, what happens? The seeds are formed, but without fertilization. So here in this case, there is an assured reproduction. It is asexual reproduction, assured asexual reproduction, but without the pollinators. So it's an asexual reproduction. It is an asexual reproduction. Here it can be formed from the new cells, it can be formed from the egg cell, but without fertilization. Diploid egg cell without fertilization. New cells also, like that. So Parthenocarpy and polyembryony. One more thing is that polyembryony, where many number of embryos are formed. Here many number of embryos are formed from the new cells. It's also called as nucellar polyembryony. Many embryos are formed around the embryo sac. It is present in citrus. Polyembryony can be observed in citrus. Each ovule contains many embryos. Then occurrence of more than one embryo in a seed is referred to as polyembryo.